My name is Aryeh Batt. I'm the co-founder and CEO of Precise Bio. Precise Bio is a regenerative medicine company, 3D printing of tissues for transplanting in humans in places where there are not enough tissues available worldwide, or in places where replacing a damaged tissue can solve a therapeutic need. Precise Bio is developing and bringing to the clinic tissues and organs that are biofabricated, where we are using a unique 3D printing technology that can mimic the anatomical structure as it is in the human body. We are aware of various trends in healthcare that when we look 10, 20 years forward, we will see that these trends are going to be materializing. People are living longer. The balance between older people and younger people in the population is changing. We are seeing that healthcare is moving to personalization, where we are taking care of each patient and suiting the therapeutic methods to the person himself. We are aware of artificial intelligence, Internet of Medical Things, big data. And I would say that also through the COVID-19, we are aware also of remoteness that is taking significant use in healthcare. And we are also aware of biofabricated tissues that when we look 10, 15 years forward, this will be something that will be very common. Precise Bio is exactly in that field of biofabricated tissues. Our vision is, and here Precise Bio leads the path to a world where there will be less need of donor tissues and organs. The waiting list that we are aware today of for certain types of organs and tissues will not exist. And these technologies can address also life-saving needs, but also life quality as vision, hearing, orthopedics, cardiology, and others. I would say that 15, 20 years from now, we would also see biofabricated kidneys and other complex organs. The biofabrication starts with a 3D printer that has a very unique technology. We are using a laser printer that essentially can deposit cell by cell. This accuracy of printing cell by cell is very important when we come to mimic the natural tissue, because this way we can copy the exact anatomical structure as the tissue has in the body. And if we fabricate our tissue in this manner, then we will be able to receive a tissue that will be adopted by the body, and it can replace the damaged tissue that is currently in the body. Essentially, what Precise is doing is taking 20 years of regenerative medicine technologies that have been developed, technologies as cell technology, cell expansion, stem cells, biomaterials, and bringing it together with 3D printing, which enables us to fabricate tissues and organs for transplanting and bringing them to the clinic. If we look at 3D printing, so 3D printing has also been evolving over the years. And I would say that here, Precise is not inventing anything. And in the same method that we can print this vase here in the middle, take a 3D image, reduce it to 2D slices in the computer, and then print layer by layer, a similar approach can be done for a biological tissue. We have started in the field of ophthalmology, and it can be very easy understood. If we look, for example, at the cornea, and we think of the process of a 3D printer putting down layer by layer, then if we look at the structure of a cornea that is essentially layer by layer built, where we have the endothelial layer and then the descendant membrane, the stroma, the Baumann layer, and the epithelium, it's very intuitive to think of a 3D printing that can fabricate this type of tissue. The process, and what we call it Precise Bio or 4D process, starts from the cells. The source of the cells can be from stem cells, but they can also be from primary cells. We can take a cadaver cornea, isolate the cells from it, then expand them, and we can expand them 70-fold, 100-fold. So from one cadaver cornea, we will have enough cells to fabricate 70 or 100 corneas. Then we prepare the bioink, which the bioink essentially is a collagen-based material which mimics the ECM, the extracellular matrix of the tissue. We bring these together, the cells together with the ECM, and then we can biofabricate the tissue in our 3D printing technology. And this will structure essentially the tissue as I mentioned, in the same anatomical structure as it in the body. But this isn't the end of the process. After we have the cells in place, we still have to mature this tissue to function as a tissue as it is in the body. And this essentially is our fourth dimension, the time where we have a maturing process in a bioreactor. And after this process, just for example, for a cornea, it can be 2 to 3 days, we will have a mature tissue ready to be transplanted in the human eye. At Precise, we take these technologies from theory to practice. And I'll just bring several examples of tissues that we have already fabricated. We are at the preclinical study stage at this time, and we anticipate to reach the clinic in 2022. I will show several examples of tissues that we have biofabricated in our labs, transplanted in animals' eye, and they function. The first one is the cornea graft, and you can see here at the bottom a rabbit's eye that has a disease. Essentially, we mimic the disease by removing the endothelial layer. And here in the middle, you can see the tissue as it is transplanted in the eye with a follow-up of 3 weeks here. And at the right side here, you can see the endothelial cells as they are in the rabbit's eye, where we are talking here about a very nice cell density of 2,400 cells per square millimeter. And the result we can see here at the top, where we have a follow-up of the rabbit's eye after we transplanted the tissue in the eye, where we can see the functionality of the endothelial layer that was transplanted in the rabbit's eye. Another example here is an intrastromal lenticule that we have transplanted inside the stroma of a rabbit's eye. We make the pocket with femtosecond laser, and then we transplant the lenticule inside the stroma. And also here we can see a follow-up of 3.5 months where the lenticule maintains stability over time, it maintains transparency, and essentially we can address and use this lenticule also for keratoconus, but also in the future for elective surgery as vision correction. The third tissue that I would like to present is a retinal tissue. The lenticule, as transplanted in the rabbit's eye, can also be shaped by a standard laser. And here we can see that we can shape the laser. This is the tissue that we have fabricated, and we can shape it for the specific patient and the specific patient's needs. What is very important, and this is every surgeon will say, that the handling of the tissue is very important for the success of the transplant, the optical acuity, of course, as an outcome, and the fast recovery of the patient. And essentially, when we fabricate the tissue, we fabricate for these 3 parameters. And you can see here in this example how easy it is to handle. We're showing here a 20 micron layer, where you can see that the mechanical properties really enable us to do whatever we want with the tissue. When we put it back into the eye, it will just open up. And here on the right side, you can also see an example where we are using a standard tool, and the tissue will just open up in the eye as surgeon transplants it in the eye. We have done studies and we have shipped these tissues around the world, from the US to the Middle East and back, and then transplanted it, and we were able to maintain the cell density, the mechanical properties, and essentially we can ship these worldwide in a relatively standard shipment, and they will be essentially available worldwide. The third tissue that I would like to show is a retinal tissue. This is still at early stages, where what we are seeing here is a RPE layer of the retina, and we have practiced this on various scaffolds. We're talking here about a scaffold that is very transparent. It maintains the cell viability and it holds the cells intact in a very controlled method. And you can see here where we have done here a transplant, a subretinal transplant of this tissue inside bovine eye. This was done with a unique tool that we have developed in Precise Bio, a tool that was developed together with surgeons And this is we have exercised these transplants with this tool. This currently is still at ex vivo, and in the middle of June, in the coming weeks, we will be already doing an in vivo study with this RPE layer. These are just several examples of tissues that we have fabricated or biofabricated with our 3D printing technology and other printing technologies at the Precise Labs. But in the future, just in ophthalmology, we will be seeing other tissues that we'll be able to fabricate. We're talking about the ocular surface, where we can address limbal stem cell deficiency, the corneal EK, endothelial keratoplasty, which we have exhibited already in preclinical studies. We will be able to, in the future, also fabricate lenses. We're talking about intrastromal lenticulae for various indications, retinal tissue, and essentially the sky here is the limit. We have the cells, we have the technology. All of these technologies can be brought together, and in the future, we will be seeing other tissues that will be biofabricated with 3D printers and other printing technologies. Thank you.